Hello, my name is Sarah Neal Webb, and I am the 2018 American Society of Primatologists Primate Welfare Award winner. As part of this honor, the research that I presented is being highlighted on the ASB website, and so that's what you're seeing here. So I am, without further ado, pleased to share this research project with you. So it was titled, Does Group Size Matter? Captive Chimpanzee Behavior as a Function of Group Size and Composition. And so we'll start with the introduction. The National Institutes of Health, or NIH, recommends that captive chimpanzees be maintained in multi-male, multi-female groups of at least 7 animals as part of an ethologically appropriate environment. Furthermore, the Association of Zoos and Aquariums, or the AZA, in their Chimpanzee Care Manual published in 2010, recommends that chimpanzees be maintained in age and sex diverse groups that meet or exceed 3 males and 5 females. These recommendations are based on the idea that wild chimpanzee societies and environments represent the gold standard after which the captive chimpanzee environment should be modeled, and that duplicating the group sizes and compositions of wild chimpanzees will result in the highest level of well-being for captive chimpanzees. However, many suggest that natural conditions and behaviors should be simulated through functionally appropriate captive environments, or FACEs, which promote opportunities to engage in species-typical behavior through functional simulations of natural conditions. These FACEs focus on ways that the environment functions to elicit natural behaviors rather than placing the focus on replication of features of the natural environment. The recommendations for captive chimpanzee group size and composition are based primarily on data gathered from wild chimpanzees that live in large fission-fusion societies of up to 120 individuals per community, with an average party size of about 5.7 individuals. However, wild chimpanzees live in such social systems because they have to adapt to the environmental pressures present in their immediate environment, and captive chimpanzees don't necessarily face these environmental pressures. As such, some have posited that natural wild conditions may not be the most relevant metric for defining optimal captive group size and composition. So we aimed to empirically examine group size and composition recommendations in an effort to provide additional data for use in defining captive chimpanzee FACEs. So we collected data on a total of 120 chimpanzees in 17 separate social groups ranging in size from 4 to 10 individuals per group. Chimpanzees at the time of the study ranged in age from 15 to 56 years of age, although the mean age was about 31 and a half. We used 15-minute continuous focal animal samples to collect behavioral data on the chimpanzees for a total of about 713 hours of behavioral data. We used a fairly standard ethogram that included locomotive, aggressive, affiliative, abnormal, and inactive behaviors, as well as rough scratching, which is a behavioral indicator of anxiety. We also calculated behavioral diversity scores, which represented the average number of different behaviors that each chimpanzee exhibited across the study period. We lastly calculated a proximity score for each chimpanzee, which represented the ratio of time spent near or touching social group mates. Our predictor variables, or the variables in which we were interested in examining in relation to behavior, were group size, dichotomized into 6 or fewer versus 7 or more, within group age range, which was the youngest animal's age subtracted from the oldest animal's age, and this was categorized into small, medium, and large age ranges. Lastly, we also examined percentage of males within the group, which was dichotomized into less than half males within the group or at least half males within the group. That actually also turned out to be a median split, which worked out well. So each of these 3 predictor variables was put into a Mancova with average age of the group as a covariate. So if we look at each of the effects in a little bit more detail, first we see that there was a main effect of group size for locomotion, such that chimpanzees in larger groups showed higher levels of locomotion. We also saw that locomotion increased as within group age range increased. And again, remember, this is not age of the chimpanzee, but within group age range. So the youngest animal's age minus the oldest animal's age. There was a significant interaction effect for locomotion between group size and the percentage of males within the group, such that within groups that had at least half males, levels of locomotion were about the same across group sizes. But within groups that had less than half males, locomotion was about 4.5% higher in groups of 7 or more compared to groups of 6 or fewer. We also see that the effect of increased locomotion with a higher age range is more pronounced in larger groups. And as an example of some locomotion, I've included some videos here. Here we see some locomotion in a group of 7 with less than half males and a within group age range of 6 years. So this would be classified as a group with a small age range. Here is some grooming at the top of the cupula here. We also see some locomotion. This is in a group of 6 with less than half males and a large age range, 21-year age range. Lastly, we see an interaction effect for affiliation, wherein within groups with less than half males, affiliation is similar across group sizes, but in groups with at least half males, affiliation is higher in larger groups. I also have some videos of affiliation here. Here we see some grooming, some affiliative behavior in a group of 9 with less than half males and a 12-year within group age range. So this would be classified as a group with a medium age range. Here we see some grooming and resting. This is in a group of 6 with less than half males and a large age range. So what does all of this mean? What can we conclude? So first, we found that locomotion differed as a function of age range, as well as a function of the interaction between group size and percentage of males within the group. We also saw that affiliation differed as a function of group size and percentage of males. So taken together, these data do suggest that there are certain advantages to housing captive chimpanzees in larger, more age-diverse groups with more males. As such, this does provide some support for the current NIH and AZA recommendations. In the context of functionally appropriate group sizes and compositions, there may be some room for welfare enhancement by increasing locomotion and affiliation through increases in social complexity. The higher percentage of locomotion within groups with a wider age range, as well as the higher percentage of affiliation in larger groups with more males, may be an indication that larger, more age-diverse groups with more males possess higher levels of social complexity. This complexity may require increased locomotion and affiliation in order to maintain social bonds as chimpanzees move between and among group mates to socialize. These findings can be applied in a variety of captive settings. For example, careful construction of group size and composition could be one way to ensure a higher amount of locomotion within a group, thereby perhaps reducing chances of obesity in the long term. However, it's important to note that these data seem to be a bit more limited in their application to existing social groups. So overall, the data seem to point to some advantages of larger groups with a wider age range and more males, but it's important to point out that we found a relatively small number of behavioral differences across different group sizes and compositions. This suggests that other group sizes and compositions, for example, smaller groups with perhaps a smaller age range or fewer males, are still functionally appropriate. Levels of aggression, abnormal behaviors, and rough scratching, which is a commonly used indicator of decreased well-being, none of those were significantly different across groups in the current study, and levels of each of those behaviors were low across all group sizes and compositions, comprising less than 0.5% or 1% of all activity. Lastly, the overall levels of affiliation, inactivity, and locomotion across group sizes and compositions, while they were statistically different in the current study, were all within ranges reported in other studies of captive chimpanzee behavior and are also similar to ranges reported in the wild. Therefore, the various group sizes and compositions found in this study seem to be successful in promoting species-typical behavior, and as such are, or do seem to be, functionally appropriate. The last thing I'd like to point out is that a manuscript detailing this data, as well as some additional data, was recently submitted for publication, so you can keep an eye out for that in the future. Lastly, I just want to thank my advisors and committee members, my co-authors Steve Shapiro and Yan Hao, as well as Nancy and Otto. A special thank you to my colleagues Mary, Michelle, Susan, and Lisa, as well as the National Center for Chimpanzee Care staff, and of course the chimpanzees. So thank you all for listening.